Hello, AP Bio. Here is your second half of your prokaryotic gene regulation notes. All right, so yesterday we talked a little bit about the basic idea of how we can regulate genes, whether those are inducible or repressible. All right, so today we're going to look at more in depth at each of those, both a repressible and an inducible operon. Okay, so to remind you, our operon, okay, again, is an operator, a promoter, and the genes that they control. Okay, so there's different ways that we can. Oh, shoot. Ah, okay. Let me go back. There we go. So let's look at those different parts. So we have the promoter region, which we're familiar with, right? In that promoter region, we have the TATA box where the RNA polymerase attaches. But in addition to that, we have the operator where the repressor can attach or detach. Okay. So that repressor, if it's present, basically shuts off that gene because the RNA polymerase cannot move forward. Okay, if that repressor is gone, RNA polymerase can cruise along. And what happens is, there we go. Then we can make that, transcribe that mRNA. And what's another thing that's unique about an operon is all the genes for a metabolic pathway are all lumped together so that we make all the enzymes needed at the same time. And then those can just get to work doing what they need to do. All right. So we can have transcription, translation of all the enzymes for a metabolic pathway. Boom, 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 right? Right. All of them occurring right there. Okay. So very efficient. And since we don't have introns, it can be very quick as well. All right. So let's look at two of those. First one, the repressible operon. Okay, we use tryptophan for that example. Okay, we need tryptophan to be made. It's an amino acid, so it's required to help build amino acids, or I'm sorry, help build proteins in the organism and help make whatever. I'm rambling. Make the proteins. Okay, so here we go. We have our promoter region. We have RNA polymerase attached. We have the operator with the repressor. We have all the enzymes to make tryptophan. So typically, this is turned on. We're making tryptophan. Okay, tryptophan is absolutely required. It's one of the 20 required amino acids. Okay, so typically the prokaryote is just making those enzymes and we're making tryptophan. However, sometimes we might we've made plenty. It's done its job. It's making all the proteins that are needed. And we have this excess buildup of tryptophan. Well, if a bunch of tryptophan's floating around, some is bound to bump into the repressor. And if it does, if it bumps into that repressor, it will change the shape of that repressor so that it can bind with the operator and basically turn off the gene. Okay, if we have lots of tryptophan, maybe some of the tryptophan comes out of the repressor, but then another will bump into it and it'll keep it off, okay? But if we use up all the tryptophan, eventually that bit that comes off the repressor will not be replaced. The repressor will release and will turn back on tryptophan until excess is made and then it's repressed again, okay? So that tryptophan conformationally changes the repressor protein. Okay, so it's kind of like an allosteric molecule, right? On an other site, not the active site. But it's not an enzyme, it's a repressor, right? So it allosterically changes the conformation so that it will fit with the operator. All right, and so a closer look just at the repressor protein again. Oh, we have excess tryptophan. It attaches to the repressor, changes shape, turns off that operon. Okay. Then we save a bunch of energy because we are no longer making these enzymes that we don't need. All right, on the flip side of that, there are inducible operons. Okay, so lactose was our example of an inducible operon, meaning it's typically turned off, but in the presence of something, it can be turned on. All right, so prokaryote is living its life, doing its thing, and then all of a sudden, boom, there's tons of lactose around it. Well, we need to take advantage of that lactose, break it down for energy. So if lactose is present, some of it will bump into that repressor, change the conformation of that repressor so that it releases from the operator. RNA polymerase is let loose and it can go down. Okay, so we have our operon or lactose attached, repressor releases, and boom, we can transcribe the genes in that operon. They all turn into enzymes and we're able to break down lactose. Okay, so that conformational change again, but this time it's releasing, it's releasing that repressor. Okay. Okay, another look at that. You can see, usually it's attached. It has that shape so that it can attach, but when lactose is present, changes its shape so it's released. Boom, we make that mRNA that codes for the enzymes to break down lactose. Okay, lactose again is an allosteric regulator, meaning it attaches to the other side of that repressor protein. Okay, so lactose, the lac operon was actually the first operon that was discovered, and now we've found a bunch since. All right, so summary here. Repressible operons, we usually use those for anabolic processes, okay? So these are things that are typically needing to be built, so they're on, we're building what we need, but if we make excess, okay, we repress those, we can turn them off. The camper. Sorry, my dog's being a wuss. All right. So inducible are going to be things typically that are catabolic pathways. Like all of a sudden something's present, we can break it down for nutrients. Well, we need to induce that operon, turn it on so that we can break down that food and get energy. Okay, but we don't want to waste energy making those enzymes if that food's never present. Like if lactose is never around or when it isn't around, we don't want to be wasting energy making those enzymes, right? And so it's not until it is around. And prokaryotes really need to be able to react quickly. And so these repressible inducible operons work well for that. Boom, something's present. Turn it on, let's get energy from it and make all those enzymes. All right, so those are the basics, and I'll give you some imagery of some of that in the near future so you can kind of see that happen. But those are the basics of the operon idea and how prokaryotes regulate their genes. Please let me know if we need clarification on anything or you have specific questions. All right. Have a great day, and I will be posting more in the future.